Again, welcome to today. First off, let me start with anything from the last couple days not clear, anything we should review that you don't feel comfortable with or you don't quite understand. Is there any questions you'd like to start with just to come up to speed from the last couple days? Is there anything like that? Anyone? No? Okay, so I must be doing a pretty good job, right? Okay, so Confocal Microscope, which is the instrument you have here. It's the, I'd say, you see them in a lot of places. I mean, they've been around since about maybe 1989, 1990 it started. I never really got to work on one until probably mid-2000s or so. They're definitely becoming more common. You see them more often. It's a pretty standard research instrument. It is awesome. I mean, it is an awesome tool. It does a lot of stuff. I mean, it's very powerful. It can do many things. But you know, a lot of times it's sort of too much. Like we talked yesterday about wide field and such. I mean, if you're just, looking at a cell to see if it's green or to see if it's red, do you really need to go to the effort of getting the confocal and working on it just to say, oh, my cell's green or my cell's red? I mean, so I guess the thing to say is you always want to start simple. I mean, just like when I teach, I try to keep things simple and then we build to something more complex. So I think the way to go about it is try to answer your questions always with something simple. It'll make more sense to you, it's just easier, and oftentimes it's less expensive. But when you do need the confocal or a higher type of microscopy like I'll talk about today, I mean, sometimes you just have to have it. But remember that the simpler things a lot of times in life do work really well. Okay, so what we talked about yesterday is wide field microscopy, and I showed you some of those pictures where I deconvolved the cells and they looked really nice. And wide field works great on cells and thin pieces of tissue because it works on things that are thin. Because a little bit we talked about the idea of how the light, because if something's thicker, the light's going to go into it and the light's going to get bent by all the molecules in there, all the carbohydrates, all the proteins, all the fats. The light's going to get scattered in numerous directions. And with the wide field, you're going to get more blurring because everything's getting bounced all over. The light's getting bounced in many directions. Whereas a confocal, when we talk about it a little bit more, it tends to get rid of that scattering to some extent. Okay, so it'll make a sharper, cleaner image. So thin stuff, I mean, I have no problem working on the wide field. Cells especially, because cells obviously are very thin, just a few microns thick. But if you're going to do tissue, you're going to go deep. You're almost for sure going to need some type of confocal. So this is the classic confocal, non-confocal image. The top would be wide field, it's a cell, and the bottom would be a confocal image. So you definitely get that better resolution. And what have we been talking about since we started this course? Resolution, right? And what is everything? NA, because NA drives resolution, okay? NA drives resolution, and resolution is what it's all about. You have to be able to tell two parts apart from each other, two separate entities. You've got to be able to distinguish one from the other. Okay, so just take a minute and read this. I found this in a book, and I just thought it was a very well-written, succinct description of what a confocal does. So take a couple minutes and take a read of that and think about it a second. Okay, so again, it comes back to the idea of what we briefly talked about yesterday, how if we're looking at this from the side, and this is what is in focus, right? A wide field microscope, the light comes in, the light from above it, the light from below it all goes to the camera or to your eyes and becomes part of the image. Even this part that's not in focus. But with the confocal, by the use of a pinhole we talked about yesterday, remember if you go like this, you look through it, things are a little cleaner, a little crisper. They just seem a little better because you're not bringing in all this extra light. Same idea here. You're not bringing in all this extra light that's not in focus. You're just bringing in that thin focal plane. Okay, that is the basis of confocal microscopy. Okay. And as it says, a confocal microscope selectively collects light from a thin optical section at a plane of focus in the specimen. Structures within the focal plane appear more sharply defined than with that of a conventional microscope because there's not as much out of focus light. Okay.
Can you explain what is in the optical section?
Okay. That's exactly where we're going right now. So next couple slides will show what that means. Because think about it, when we look at a piece of tissue, like say you have a piece of liver, and if you want to look at it regularly, what do? You cut it, right? Like we talked about the other day, you have a cryostat to cut a thin section, or you have the microtome to cut a very thin section, and you take that section, put it on a slide. Take the next section, put it on a slide. Take that next section, put it on a slide, right? And that way you can look through the tissue physically. You could literally cut it and look at each piece, and that way you could go into the tissue and look at it that way. But here, instead of cutting it, we're going to use light to do our cutting for us. Optical sectioning. Now, this here is a CAT scan, okay? And I don't know how many of you have been hurt or injured or have had different things done. My daughter had a seizure a few months ago, so what they did is they did a CAT scan of her brain. And what a CAT scan of the brain is basically is they do a section, like an X-ray, 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 and they build those up, and they're able to get then a three-dimensional reconstruction of her brain, okay? So you've all heard of CAT scans before, okay, or CT scans, I guess they call them too. Okay, same idea here, except instead of using X-rays, we are using optics. We are using a microscope to do this, okay, on obviously a much smaller level than a brain. Okay, so if we take a look at this here, okay, so we have our piece of tissue represented by a ball, more or less, or a sphere in 3 dimensions, a sphere or a ball. So we have it on the confocal microscope. So the first thing we do is we take an optical slice. And what I mean by an optical slice is we take a plane through there. We take that one optical slice, that image. We focus on that green image right there, that green flat plane. And then what we do is we use our microscope, we move down to another plane, to the next plane down. And we keep this picture. Then we move down. We do another plane. We keep that picture. We go to another plane. We go down. We keep that picture. And then the final optical slice is scanned and stored in the computer. So now what we have from that ball is we have a series of images that represent the ball. And as you can see, the top image is just, the little circle's not as big because you're at the top of the ball. The middle ones are a little wider. You're in the middle of the ball. And then the bottom one, again, you're like at the lower end of the ball. Well, you could do the exact same thing physically. You could take that microtome and you could cut that ball. You could physically cut that ball, put the pieces out in order, and you'd see how that ball is put together or how that structure is put together. But here, by using the confocal microscope, we're able to take that tissue apart optically, but not physically. Okay, so we're able to, yes?
Does it matter how many planes we take?
It does to some extent. We'll get into that in a little bit. You can really take as many as you want. But the problem becomes, if you take too many and they're, okay, so what it becomes is this. Let's say you take a plane, and we'll say the plane's this thick, okay? Now, if I took the next plane here, I would miss this, okay? So I need some overlap in my planes so I can get a clear picture of the whole object. Like I need some type of overlap. But if my overlap is like this, like very, very, very high, almost the same, then it's too much overlap. You're like almost overkill. You've generated too much information. So the nice thing about these confocals, the way they're designed, is it's big physical formulas, you know, big math formulas to determine, hey, how much overlap should I have? How far should I move in microns to get that overlap? But the way these confocals are designed now, when you use a certain objective and you tell it what wavelength and stuff you're going to use, it'll automatically calculate that for you, and it will give you the appropriate step size. And that's what that's called. That moving from one plane to the next plane to the next plane is called step size. And sometimes you'll hear me refer to it as Z step, because what I'm doing, remember, I am moving down in Z. Okay? So yes, you have to have some overlap, like your question was, but not too much. But the confocals today, the math is done for you behind the scenes. And a lot of times you'll see something come up on a screen that says recommended step size, 2 microns or 1 micron. And the thing about that is the higher the magnification you use, the smaller your step size is going to be. Because remember what we talked about the other day. At 10x, this much is in focus. At 20x, this much is in focus. 40x, this much is in focus. Okay, so obviously if you have something this thin, you're not going to be able to move real far to get that overlap. So the higher the magnification, the tighter the step size. Okay, does that make sense? I mean, it's an important concept. That was a very good question. Okay, so as we can see though, we now have these numerous pictures of the ball, and by using software and stuff, we can put these all back together and generate a picture of the ball. Okay, so we have optically cut the ball. Okay, so what we call those is basically a Z-stack. Anytime you have that set of like that, that's a Z-stack. Okay, and when you take that Z-stack and you put it on the computer and you look at it, we call those projections. Different projections because you can look at it from different angles. You can look at it like, you can take the ball, you can look at the ball from the top. You can take the ball, you can look at the ball from the side. Okay, you could look at the ball from the bottom. Okay, you can look at the ball numerous different ways. Those are all called projections, just depending on the angle you want to look at the ball to allow you to get the information you need from it. Okay, and like we just talked about in terms of the overlap, that's called the step size. So anytime though we take those different planes and we put them back together in 3D to rebuild the ball, we call that a reconstruction or we call that a rendering. Okay, so if you hear me use these terms, projections, which is looking at the ball from different angles, reconstructions, I mean, think about it, it's a perfect word for it. We have reconstructed the ball. We have made the ball whole again, okay? Or sometimes instead of reconstructions, we call that a rendering. Okay, and again, the step is called a Z-step. And the way I think of it and the way I tell people to think about it is something that's common, I think. I haven't seen anybody doing it here yet, but I'm guessing you probably do this in India too, is it's like a deck of cards. Each picture is basically like a deck of, you're taking a deck of cards, the top card, the next 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 card. So it's almost like each picture, and then if you put the whole deck of cards back together, you build the picture back together. So you do play cards here? I'm guessing it's probably big. It's big everywhere, I would think. Okay. 52 cards in your deck? Is it the 54? Oh, the jokers. Okay, so we all have the same cards. Okay, okay, good. Okay, a deck of cards. So, again, to reiterate this idea, what is optical sectioning that Madam asked? So basically what it is, it's able, again, instead of a ball here, we're looking at a little bit different shape, but the idea of taking the top, taking the next one, taking the next one, taking the next one, and building it back together. So what we call the physical cutting, on the cryostat, we call that cryosectioning. Here, we call this optical sectioning. Okay. Okay, and the nice thing about it is here, we're looking at an optical sectioning image on the right because this is a very thick specimen here, 100 micron thick sea urchin embryo. And by optical sectioning, we can get rid of a lot of that blurring that we see in a wide field microscope. The problem here is there's just too much blur. There's too many things in the way. The light is scattered, the light is refracted, the light is bounced all over. It's hard to get information out of it because it's not clean. So again, we're going to take advantage of that pinhole to try to get rid of a lot of that out of focus light and just collect the focused light. Okay, so with the confocal, you can literally take a cell, something that small, that's that thin, only a few microns, and you can literally cut that cell into numerous parts and rebuild that cell in 3D. Okay, so it's a very powerful tool in modern biology. Okay, and in numerous other applications. A lot of times I work with engineers looking at particles suspended in like a liquid or a solution or something. And a lot of times we'll use the confocal to try to determine what the structure of that is. So it's not just biology, but biology just absolutely loves this technique. Okay, so here's a classic, and hopefully we're going to try to look at this today. I actually brought a slide of a pollen grain. So, I mean, when you look at a pollen grain, it looks basically like a ball with a bunch of spikes coming out of it, more or less. I don't know if you've ever seen a picture of one. But here, you can see if you use the confocal, you'd start with our deck of cards. Card number 1, the top. Card number 2, card number 3, working your way in, and eventually you could take all these and you could build back the whole idea of the pollen grain itself, okay, without actually having to take it and physically cut it. Okay, this actually, I think I talked about this earlier the other day. This Olympus Micro website's a really good website. It explains this stuff, and it's also has a very nice explanation of what we're going to talk about a little bit, co-localization. And I know a lot of you mentioned in your that questions I put out there, those cards, a lot of you want to do co-localization. So I would definitely look at this Olympus site and the Nikon microscope site. Both have very nice explanations of co-localization that go beyond what I'm going to teach you today. So if you want more information, there is literally a ton of stuff out there on the net. And at the end, we'll talk about the 4 big microscope companies, but their websites are very nice and very educational and very well done. Okay, so we've been talking about cells. We can do it in cells. But this one, we can also do the confocal and tissue. Now, this is my favorite tissue I work with all the time, which is retina in the eye. I took these pictures on the confocal. So what we're going to do here is we're going to look at blood vessels in the retina. Okay. See, they're starting to come into focus. We're working our way in, working our way in, working our way in, working our way in. We start seeing the vessels. Okay, and then they sort of come a little more, and I think it disappears here, if I remember right. Okay, but what we're doing is we're just working our way into the retina. Okay, let me play that one more time. So we're just, taking a Z-stack and going into the retina. Okay, you see a vessel, then it disappears. You see another vessel, then it disappears. You see some more vessels, and it disappears. Okay, so if you took any one of those single cards, there's probably just, you have a little bit of the retina there, but not a whole lot. So what we do is something called maximum projection. What we do is we take that deck of cards, okay, and basically we smash that deck of cards into one card. How does it do this? What is the way it does this? Well, here's the way it does that. It takes every card, okay, and what did we talk about yesterday? We talked about pixels, right? So what it does is it takes pixel number 1 up here in the corner. It goes through the whole deck of cards, and it says, out of the whole deck of cards, every single pixel number 1 in the deck of cards, which pixel is the brightest? Well, maybe it's in card number 12. Maybe it's in card number 16. Then it keeps that pixel. It goes to pixel number 2. It does the same thing. Which pixel number 2 in all the deck of cards is the brightest? So what you get is you get basically, by doing that and doing it on every single pixel, you get a one card view of what the image representation is. Okay, so we call this a maximum projection. Okay, and other companies you'll see they'll call this extended focus. So those are like the 2 terms they use for this idea. But what it is basically, again, it goes back to math. It goes back to take the pixel, find the brightest. Take the pixel, find the brightest. Go through the deck of cards, find the brightest. Okay, does that make sense? Okay. Okay. So you could also have taken that deck of cards, and you could have made it into like a 3D rendering, and you could just take a single picture of that too, like just a single picture that's sort of like in 3D. You can sort of see how these vessels sort of come at you a little bit, or sort of go away from you. So you can take a single, so you can also make like a single card that sort of represents the 3D of it. Okay. Okay, but the nice thing about confocal is this. We take our cards, and maybe we use 2 colors in our cards. So we have 2 colors of cards here. But the cool thing about it then is we can look at it in 3D. So we can really appreciate what the, what the true, I guess, truer structure is. So what I'm doing is I'm spinning around the retina here. I'm looking at it from different angles in the red. And now I'm going to add my green in, which was a different molecule. And then I'm able to look at the relationship of both of them in terms of 3D, how they relate to each other. Because what it is here, what we're actually looking at is we're looking at the red. This is a diabetic retinopathy mouse. Okay, so in diabetes, what happens is capillaries tend to degenerate and die in the retina. So what we're looking at here actually is red, because of the way we did it. Red are actually dead capillaries that signify they're gone, they're dead, whereas the green are actually capillaries that are still alive. The way we do this is we take a fluorescent molecule, and the green fluorescent molecule, we inject it into the tail of the live mouse, into the tail vein of the live mouse. So it perfuses throughout the mouse. And obviously, it can't go into dead vessels because they're not functioning anymore. They are closed off. So what we do is we basically have then green shows where all our live vessels are, and then we go back in with the counter stain that just stains the vessels in general. So by subtraction, it shows us the red then is the dead vessels, the green is the live vessels. Okay. So pretty cool technique. I mean, something, things you can think about with fluorescence. I mean, because fluorescence doesn't just show live things, you can use it for dead things. You just have to think it through. What do I want to look at? How do I want to look at it? Use your 2 colors in an effective manner, or 3 colors or whatever. Okay. Okay. So co-localization. Co-localization is a big part of confocal. As soon as you say to me, hey, I want to do co-localization, then first thing that snaps in my mind is, okay, well, we're probably going to have to go to confocal. Almost for sure. Okay? The reason being, again, I think I did this yesterday, but to reiterate the point, I have protein A, right? Protein A is here. I have protein B. Well, obviously here they're not co-localized, right? Protein A is over here, protein B is over here. But on a wide field microscope, if you're looking at it, you'd say, oh, okay, here's protein A, and I'm looking at it from where you're sitting. Here's protein B, and my gosh, it looks like they're in the exact same place. So you would say they're co-localized. But by being able to cut this into 3 dimensions, into optical sections, actually, if we look at it then from the side, we realize that protein A and protein B are in different places. They are not co-localized. Okay, on a wide field, it would say yes, they are, but in reality, they are not. Okay. Okay, so let's see what we wrote here. We will focus on how the analysis distribution of stimulus fronts can be used to determine 2 probes. So basically, you're looking at 2 different colors. And again, remember what else we talked about yesterday. You don't want to use 2 colors that bleed into each other, that overlap with each other. Anytime I hear co-localization, I like to use a green, if I can, way over here. And I like to use a far red, which is way over here. Okay, I like to try to spectrally separate myself as best I can so I don't run into this bleed through. Okay, so I like to try to pick probes or fluorophores that are spectrally far apart so I don't fool myself into thinking, oh, it's co-localization when it's nothing more than just the bleeding. Okay. Okay. The one thing to keep in mind is I think people overestimate the power of what this is. Is that, okay, this approach is not appropriate for detecting molecular interactions. Okay, even in a super resolution system. Okay, just because 2 proteins co-localize doesn't mean those 2 proteins are working together. Okay, it doesn't mean that. Because you could have 2 proteins co-localizing in a complex. Like you could have a rather large complex. You could have a protein here and a protein here. Well, at the resolution, the ability to separate, it looks like they're in the same place. But that doesn't necessarily mean they're touching. It doesn't necessarily mean they're interacting. Yes, they are very, very, very close. And they may be touching. And they may be interacting. But it does not prove that. Okay? So a lot of times people are telling me, oh, this proves. No, it doesn't prove that. You need to use other techniques, biochemical techniques and such. And we'll talk about something a little later called FRET that helps prove that tightness of association. But co-localization a lot of times is nice for, say you have a protein. A lot of times here's what we'll see. You'll have a protein A, and you have your protein B, and you add a drug or something, and it seems like, wow, every time we add this drug, protein B runs over here next to protein A. Seems to draw an association. Then it starts you thinking, well, okay, maybe they are interacting because they're not always interacting, but when we do this, they interact. So yes, maybe they do work together. And it definitely puts you on the right track to realizing, hey, maybe they do interact. Because if you add the drug and this protein goes over here and it's not even close to it, then, well, they're not interacting. Okay, so it doesn't prove that they work together, but it definitely puts you on the direction to think they definitely may work together. Okay, does that make sense? Is that clear? Okay, so yesterday we talked about pixels. Okay, pixels, the little tiny squares, the little tiny squares in your phone, where your phone has 5 megapixels or 6 megapixels or whatever they're up to these days. Well, here, because we're working in 3D, it's not just a pixel, because it's not 2D, it's like little cubes, right? So instead of calling them pixels, now we call them voxels, okay? Because they're little 3D cubes. And the one thing to keep in mind about the confocal images also is, you know, you're going to get so much information here in terms of computer storage and stuff, because it's a, you know, 111 pixel pixel, or here it's 3D, so it's going to generate more storage. Images are going to be larger and you're going to use up more space. Okay. But pixels versus voxels. Voxels are 3D. They're basically 3D pixels in a way, I guess is a way to look at it. Okay. Okay. Now, the one nice thing about co-localization is sometimes back at home, I didn't have any. I would have brought some, but I don't know what happened to them. I gave them to somebody, and you know that goes. Sometimes you lend something out, and it just sort of vanishes into thin air. So this would happen. But if you get a bead, like to test your confocal, see how well it's working, if you get a bead, and the bead will fluorescent red, and the bead will fluorescent green, like, you know, you're moving in different lasers, you're moving in different things to take the green picture and to take the red picture. Well, if the bead has red and green on it, and you start getting images like this, where you see green, you don't see red, or you see red, you don't see green, then something's wrong with your alignment of your confocal. Because if the bead should have both colors, then they should just lay right over the top of each other, 100%. So basically you take the red picture, the green picture, and if you put these two together, everything would turn yellow, because it's showing those beads are in the exact same spot. So this is just a way to check to make sure when you're doing your Z-Sacks and stuff that everything's working well. So it's always good to have some beads, and like I said, I would have brought some, but I don't know where mine went. That happens with a lot of my things. A lot of my things get lost. My hard drives get lost, my slides get lost. I guess sometimes too generous, I lend things out and they just vanish. Okay, but anyway, it's a good way to make sure that the system's working correctly. Okay, again, you saw this slide yesterday. We talked to co-localization, but here we are again, visiting the topic again. Co-localization, for the most part, will always be, you'll see your one color, you'll see your other color, and you'll see some degree of overlap. Okay, same thing in 3D. You'll see some degree of overlap. But there's also ways you can get a little more intense with co-localization. You can do it mathematically also, but again, beyond me, just, you know, over my head. So usually what I do is I look at red, green, and I look at where it's yellow. And for me, that's usually good enough for my level of understanding to try to figure out what's going on in a biological context. Okay. But again, looking at Microscopy U website, this is Nikon's website, good explanation again of co-localization. So I encourage any of you who are going to be doing co-localization, read up on it, at especially the Olympus and the Nikon websites. Okay, again, you saw this slide yesterday too. If it's done correctly, you will not get the idea of green, red, everybody yellow. This is bleed, usually. Okay, something's set up wrong with the filters, something's set up wrong with the fluorescent molecules you used. It should not be 100%. That's not reality. Okay, it's just wrong. Nothing's 100%. Okay, of course, red and green make yellow, but not everywhere. This is bleed through of one dye into another. Okay, so this came up yesterday. Somebody had asked me a question. They work with plants or something. Now this is going on to a pretty advanced technique. I don't think you have it on, I'm sure you don't have it on this instrument. We have it on a couple, I think we have it on one at Case, maybe. It's pretty expensive, but sometimes you might have to go find it if this is your problem. Okay, there's a thing called spectral unmixing. Okay, so let's say, I guess yesterday's example was chlorophyll. Everybody knows chlorophyll is green, right? So let's say we had chlorophyll, and let's say we had Alexa 488, and let's say we had GFP. Well, they're all green. So if you look at all three of those underneath the microscope with your eyes, it's just going to be a big green mess because the chlorophyll's green, the GFP's green, and the Alexa's green. Everybody's green, and it's like, well, how do you determine what's the Alexa, what's the GFP, what's the chlorophyll? How do you pull these apart? Well, the answer is you can do something called spectral unmixing. Okay, I'm going to describe what that is. Okay, so for example, you have here, the example I picked, you have Alexa 488, which we know is green. EGFP is just a variant of GFP, green fluorescent protein. And you have the mitochondria, EGFP, these are mitochondria. Mitochondria often look like sausages, sort of, like little sausages. I don't know. You guys probably don't eat sausage, do you? Sausage, you do? Okay, so you get the idea, sausages. And actin, which is the filaments here, are green. Well, here you can tell the difference just because of the shapes of them, but it's a mess. Agreed? It's just a mess. Everything's green, it's sort of blurry, it's a mess. So what we're going to do is we're going to spectrally unmix these. Okay, everything looks green. Now what do we do? Okay, so what it is although Alexa 488 is green and GFP is green, they have different spectral properties. Okay, their spectrums, if we look at it, you know, like in a spectrophotometer or whatever, they actually have a little bit different properties to them, their spectrums. Their spectrums are distinct. Alexa has one spectrum, whereas EGFP has one spectrum, whereas chlorophyll has one spectrum. Even though they all look green, and they are green to our eyes, they're actually spectrally different. So what these kind of setups can do on a microscope is you can take the unique spectrum of each one of these. So you'd come into the lab and you'd have one with you'd have a slide with chlorophyll, you'd have a slide with the EFGFP, you'd have a slide with the Alexa 488. Those would be your controls. You would take the spectrum off all three of those. Then you'd put your sample in based on the spectral data, okay? And then the computer program and such could unscramble who is who. So by using that computer program, and remember, everything's a fake color anyway, right? We learned that they're just photons hitting the thing. Everything's a fake color. So you use pseudo color, and now you can see that your Alexa 488 is nice and green. And we've made now, even though it's green fluorescent protein, we've pseudo colored it red. So now we can easily see the relationship between the actin filaments and the mitochondria again. So if the problem arises, it sounds like especially in plants, I would think, not that I work with plants that much, but I would assume you have so much green from the chlorophyll that this might be something you want to think about in the future. Maybe find an instrument somewhere here that would be able to do this type of spectral unmixing. Okay? Okay. It's sort of a niche-y kind of thing, but it does happen. I just wanted you to be aware of it. Like this whole class is about you being aware of things to, you know, because if you get these strange answers and you're scratching your head, how am I going to get past this? I just want to expose you to a lot of different techniques so you have a lot of different things in your bag of tricks to answer your questions. Okay. Okay. So we talked a little bit about the confocal, but how does it actually work? Okay. Again, the idea of the pinhole. Okay. It doesn't allow the out-of-focus light to become part of the image. So if we look here at this part, like the light comes in off the laser, again, a dichroic mirror. Okay, so this be your excitation. And then your emission, you can see here like the dotted ones are out of focus light and out of focus light. Well, it hits the detector, like it hits outside the pinhole, so that light cannot go to the detector to become part of the image. Whereas this solid line, that's the in-focus light goes to the detector. So here the pinhole is right here. This light is stopped from above, and the light from below is stopped. Okay, so it's just a schematic explaining what I was trying to show you with the piece of paper. Okay. The one problem with the pinhole, though, is that, think about it, when you go like this, you look through something, right? You've lost a lot of the light in the room. You've only seen that little bit of light. Well, at the microscope level, and like I think we may discuss, fluorescence isn't the brightest thing in the world, it's sort of weak. You've lost an awful lot of your information, okay, going through that pinhole. A lot of it gets blocked out. I mean, you want it to be that way because you want that thin optical plane. You want that thin optical section. But my gosh, you've lost a ton of light. So what are you going to do? So the reason then is we use brighter lights, we use lasers. okay, as opposed to what we talked about the other day, the mercury lights, the regular fluorescent lights on the wide field microscope. We need that extra brightness to get those fluorochromes to excite, to get it so we can see it, because we're losing so many photons because of the pinhole. A lot of the photons are bouncing off and can't get through the pinhole to the detector. Okay, but like we've talked about, if you do one thing good, then one thing bad happens. It's always a trade-off here. Good and bad always sort of go hand in hand. So lasers are much more powerful than the other light. So the problem here is it can lead to photo bleaching. Now, photo bleaching we talked about yesterday a little bit, how these fluorescent molecules, the electrons have to go up to this shell and fall back down to make fluorescence. They go up, they go down, they go up, they go down, they go up, they go down, they go up, they go down. Well, eventually, if you hit them with too much light, you're going to destroy those molecules, and they won't go up and down anymore, and the fluorescence is just dead. Okay, they are dead. They will not fluoresce anymore. That's called photo bleaching. You have bleached it out. Okay, so like if you ever took something colorful and you put bleach on it, right? It just turns it to white. It destroys the color. Same idea here. You're using light to destroy the color. Okay, because the lasers are tough. They're strong. Like they always tell you don't let a laser hit you in the eye. Okay, because it's not a good thing. Okay, but when we start talking about live cell imaging, you got to be careful with your lasers too, because there's the problem of phototoxicity. Okay, so basically what phototoxicity is this. Like let's say we leave class today and we go down to the beach, right? Okay, I'm going to get sunburned really, really quickly, right? Whereas you all have a little bit darker skin, you will not get sunburned quite as quickly, right? I mean, I'm going to suffer really quickly, I would think. I mean, I'm white, I'm coming from the north, I'm coming from the land of snow, and I'm going to burn. Well, when you're hitting cells with UV light or fluorescent light, you're burning them. I mean, you are basically sunburning them, okay? And that's what phototoxicity means. Toxic, light, photo, toxic, poison. You are light poisoning those cells. So a laser, being a stronger light, tends to hurt those cells much quicker, tends to cause that burning much quicker, okay? So it's something you got to be careful with. So a lot of times what I'll do is I'll back off with my light and I'll use my camera a little more powerful. Like I'll increase my exposure so I can collect more photons with my camera because I'm sending in a little bit less light, especially in a live cell application. And we'll talk more about that when we get to live cells. But that's sort of the trick is you turn the lights down, you turn the camera up. So your light going in is less, but your detector, you use it more powerfully. Okay, the pinhole. Just a little physicsy kind of thing here. The smaller the pinhole, the greater is the z-axis resolution, which means the smaller the pinhole, basically you can get better resolution in terms of z, up and down. But beyond a certain point, depending on numerical aperture of the lens, the pinhole diameter no longer improves z-axis resolution. So like anything, you can crank the pinhole down far enough that it'll help you, you'll get a nice z resolution, but eventually it's just overkill. Okay, and again, with these microscopes, the way they're designed now, usually when you say you're going to do 63x oil immersion, whatever, using this wavelength, the microscope calculates it and gives you the right pinhole. Okay, I mean, it's good to know the theory behind it, but with these modern microscopes, a lot of times these things are taken care of for you by the computer software. Okay, like I said, the Z step's taken care of for you by the computer software, and again, the pinhole size is taken care of Z resolution. But let's think about this a little bit. Let's think about the theory here, what's going on. If the pinhole is really, really big, you're really not blocking out the out of focus light. So instead of me going like this and looking at you and blocking out that out of focus light, if I'm like this, I mean, what am I accomplishing? I'm not really blocking anything out. So if the pinhole's too big, you basically have a wide field scope. You don't have a confocal. When you do this, right, you're back to taking this light, and this light, and sending it all back in because you're not using the pinhole correctly. Same idea. When you use a low mag objective, okay, like remember 10x, this much is in focus, 20x, this much is in focus, 40x, this much is in focus, 60x, this much is in focus. Well, if you're using a really low lens and it has a really deep focus, you're not really optically sectioning. You're just, sort of basically using a wide field microscope again. Okay, so that's why the pinhole's very important in terms of getting that correct, I guess you could say. Okay. Does that make sense? I mean, it's a concept. Like I said, it doesn't come into play that much because these modern microscopes take care of this for you. But I think it's important you understand the idea. Because I get people come to me like, we want to do confocal like 5x or something. It's like, well, why bother? I mean, you're not accomplishing anything. Okay, so obviously the pinhole or the confocal was a big hit when it came out. People are like, wow, this is a really nice tool, really useful. And then like any nice tool, people start improving upon it. And people come up with different ways to do it. Okay, so of course, when this came out, like I think it was late 80s. I'm pretty sure it was late 80s. It might have been mid 80s, but I think it was late 80s. Anyway, the one you have is called laser scanning. Okay, and as you saw it yesterday, if you were in a group 1, you got to see it goes bzz, 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 okay. There's another one called spinning disk. We're going to be talking about these now. There's one called Optigrid Deconvolution we touched on yesterday. Poor man's confocal using a wide field microscope, and then we're going to talk about 2-photon confocal. Okay.
But in the deconvolution, yesterday we discussed in the wide field, right? But there was no pinhole in the concept.
There is no pinhole, exactly. You're using, in a way, you're using math to create a type of optical section. Okay. So it's a type of confocal, I guess, in a way, but it's really not confocal. But it sort of falls under the same category because it gives you like a confocal-like image. Okay.
In general, if I say confocal means it is involving pinhole.
Yes.
And it is about 3D.
Yes. Yes. Correct. That's the. I guess you could say that's. Yeah. The take-home message is it involves a pinhole and it has to do with 3D. Right. Okay. So let's look at a couple of these different kinds. Whoops. Okay, laser scanning, this is the most common type you see out there. Most of the times when you go to an institution, they got a confocal, they say yes, it's probably some variety of a laser scanning confocal. Okay, so basically what you're going to do is you're going to move a laser spot across the specimen, collect the light from each spot or scan. Okay, it's like this. It's like, if you sweep the floor with the broom or whatever, you go back and forth, back and forth, back and forth, back and forth. Well, same idea. You're going back and forth, back and forth, back and forth across your image or your specimen to collect your image. Okay? So it's that idea of back and forth, back and forth, back and forth. And again, think about that back and forth, that's a lot of laser hitting that thing. Phototoxicity, photo bleaching, right? Okay. Sort of like a vacuum. You go back and forth, back and forth, back and forth until you're done. Okay, and I have a big dog at my house, so there's hair like you can't imagine. It is wild how much hair a dog. I mean, I have a 130-pound dog, big. I mean, he's like this tall, and it's amazing how much hair he can throw. Okay, anyway. Okay, so let's just take a look at the setup of a laser scanning confocal microscope. Now it's a little bit overkill, the picture, but the idea is what we've been talking about. Okay, the light's coming in from the laser, right? Goes through, comes down to the sample, right? It's going to come back up. It's going to go out through the pinhole and then back up to the detector. Okay, so that same idea we've been talking about with the pinhole. So what it does though is it basically just goes back and forth across it, and light originating from the in-focus focal plane passes through the pinhole to the photomultiplier, and that's what you actually collect. Okay. So again, you can get into the physics of it. You can go crazy with it. There's a lot to it. But for biology, you basically just need to know, hey, it comes through a pinhole. We excite at one wavelength. We collect an emission at another wavelength. And that's pretty much how it works for a biologist. Okay. Now, again, getting the idea of phototoxicity and photo bleaching. To avoid that, well, do you really need that whole cell? Like if your experiment is just looking at the nucleus, you have the ability to zoom in on these things. Why would you go way out here and waste extra time going back and forth, back and forth, back and forth when all you really need is right through here? So you could zoom in and just do the part you need, okay? Because laser scanning can be a little slow at times, and we'll talk about averaging in a little bit, and it slows it down a little more. And so again, if you're just looking at the nucleus, why would you take your time and take pictures of stuff way out here that's irrelevant to what you're doing? Okay. So another thing to think about, just take the image of what you need instead of just blasting away with lasers, just to blast away with lasers to hurt the specimen. Okay? And again, to avoid photo bleaching or to avoid phototoxicity. This one I used from the Zeiss webpage, another microscope company. There's basically 4 microscope companies. There's Nikon and Olympus, which are both Japanese companies. And then there's Zeiss and Leica, which are both German companies. There is no American company, and there's really nobody else in the world. So the Germans and the Japanese are your microscope manufacturers. Okay. So what we're going to talk about next is averaging. Okay. So you can sort of read this while I'm working on this. But go ahead and read this. It might be a little difficult to understand. I'll try to explain it a little better. But I looked and looked and looked, and this was the best I could come up with. Okay. So basically what this means is this. I'm going to take a picture of this cell here, right? And I'm going to use my laser, go back and forth, back and forth, back and forth across it, right? But I'm going to have parts of my cell that are real. So like if I take a picture on my first scan of it, like my first scan of it, and I see this dot, and I come back the next time, I do it again, and I see this dot again, then, and I do it a third time, and I still see the same dot, well, that dot's probably real, because it's been there all 3 of my times I've come through. All 3 of my pictures I've taken, I see that same dot, okay? So we'd probably say that's real, or we'd probably say that's our signal, because we've seen it 3 times. So if I came through this 3 different times and I see this dot here, and I come through the next time, but I don't see that dot, and I come through the next time, and I don't see that dot, well, that dot's probably noise. It was there, but then it's gone, okay? Does that make sense? So I'm taking 3 pictures. I see this one dot in all 3 pictures. Well, it's probably a real dot. But if I take all 3 pictures and I only see the dot in one of the pictures, chances are it's noise because it's not there every time. Okay? So this is the idea of signal and noise. And noise is just bad. It's just fluorescence. That's not really the truth. Okay? That's called frame averaging. You're looking at the whole picture. You're deciding out of how many averages you do, how many. Like when on your confocal, you'll see there's a thing that'll say frame average. If you put it on 4, it'll do 4 full frames. It'll look at the pixels that are positive, and if it sees it in every one, then it keeps it. If it doesn't see it in every one, it throws it out. Line, average, same idea, except as opposed to doing it for the whole frame, it's doing it across the line. So you might see one dot that you see all the time, you did it 5 times. This dot was always here. Well, this dot was only here once, so throw it away, because chances are it's not real. Okay, that's the idea of averaging. And this is the kind of quality improvement you get. You ran it once, you got this. Well, you did I guess it was a line average 8 times, so it ran 8 times across the line. It cleaned it up to this. Okay, it helps get rid of the noise. It just makes everything better. The signal pops out over the noise. Okay, so frame averaging is a thing to use to help improve the image quality. But again, like all things in microscopes, trade-off, because now you've hit the sample 4 times as opposed to hitting the sample once. More photo bleaching, more phototoxicity. Okay, same idea here, just showing an average. This one didn't turn out so well, I don't think, the picture, but the same idea of how it cleans it up. As you go through the samples, the image gets cleaner and cleaner because you're able to throw noise away. It's not, so basically things that aren't common in every picture get thrown out. Okay, so it's a powerful thing on your confocal when you have this. You have line average, you have frame average. Okay, so, yes?
If it is in case of live cell imaging.
Right. It's harder to do. I mean, you can do it, but it's definitely much harder. Because you're right, because things are moving.
You cannot draw it away because
You can do it, but you have to be quick about it. But it definitely doesn't work so well for live cell imaging. That's why a lot of times I do my live cell, I just do it on a wide field. Because the light's gentler, and mostly because the light's gentler. But you can do live cell imaging on the confocal, it's definitely more difficult. Especially if things are moving really fast, you'll have a hard time with laser scanning. So the question you're asking is going to be addressed right here, a different kind of confocal that does a better job of live cell, okay? But good question, but this is sort of the answer to what your question is. Okay, so there's actually a confocal called a spinning disc confocal. I have one of these. I think you used to use it, didn't you, Srijit? You used to use the spinning disc once in a while? Okay. So in Srijit's old department, dermatology, they had one of these, a spinning disc. Different idea. As opposed to having just one tiny pinhole, this has a disc, okay, with a bunch of pinholes. Okay. The advantage here is the spinning disc applies the pinhole across the entire image so it's faster. Okay? Because remember, with the laser scanning, I'm basically moving the pinhole, moving the pinhole, moving the pinhole, moving the pinhole, moving, you know, right? One by one by one by one. Whereas this, everything's the pinhole. So it can just take the whole picture at once. It can just go bam, 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 bam. So it's faster because it's applying the pinhole all over and it's letting you take the whole image at once. So it's faster. This is, like anytime you hear live cell confocal, you start thinking about this thing. Okay? Because it's faster. And like you said, it's live cell, it's fast. This is the solution. Usually. Okay, but always, of course, the trade-off is you lose an awful lot of light. Like I sold one of these when they first came out, and it was hard to work with because it just destroyed the light. Because think about it. You have photons of light coming through, but look at all these blue areas where the photons of light hit, and they can't come through. So you lose a ton. So the first generation of these I used to sell something called, it was called a carve, spinning disc confocal, and like we'd take it out for demonstrations and try to sell it, and like we'd look at their sample, and their sample would be very dim to start with and be like, this isn't going to be a good day. And we'd have a heck of a time trying to get it to work. But if they had a very bright sample, then we'd be okay. But for dim samples, this just didn't work. Okay. And again, physics, everything's a trade-off. Because due to the area of the disc that does not have holes and due to the physics behind it, the Z optical section, how thin you can get, you can't get as thin as with the laser scanning. Okay, so if it's important for you to be able to differentiate Z, this won't do the trick. But if you want to go fast, this is the trick. Okay, everything's a trade-off. Okay, and because again, the speed, people like this for live cell applications. Okay, I've had pretty good luck with these over the years using them for live cell. I've had some luck using the laser scanning for live cell. Just depends. You know, the other thing about cells, some cells are just a lot tougher than others. A lot tougher. And health of the cells is the main thing. Anytime we do these kind of experiments, and I don't know how much tissue culture you do, but if you're looking at your cells and you're getting ready to go do a confocal experiment, and your cells don't look like that normal, healthy look they have, don't bother. Because if they're already hurting and they're not doing well already, and you hit them with the light, they're done. So I always tell people, you know, just like the idea of garbage in, garbage out, well, if you start trying to do live cell with unhealthy cells, then you got a problem. If you got a lot of cells floating and dying, then you got a problem before you even start the experiment. So, you know, think ahead. Like I said, I've had people come over and bring me just a bunch of dead cells, like, let's go do the experiment. It's like, well, why bother? I mean, everything's dead already. So I'm not into, you know, doing experiments just to do experiments to waste everybody's time. Okay, so let's take a look at some of the architecture of how the spinning disc confocal is put together. Okay, the light comes in, you've got the spinning disc again, you've got the dichroic mirror here. Okay, and again, it's a light source coming in, your excitation, again, your emission filter, you know, using certain wavelengths of light with the excitation. emission filter, using certain wavelengths of light, and it goes through this lots of pinholes at once. But like I said, it's not my favorite because it just eats a ton of light. Okay, it's more of a niche kind of instrument. But if you're going to do live cell all day and that's all you're going to do, it's not a bad deal. Okay. But if I was just starting a lab, getting my first confocal or whatever, I'd probably go with the laser scanning, just for the quality and everything else. But if I was going to do live cell all day, I might go with this. Okay. Everything's application dependent. Okay, so let's look at another one. This one's sort of weird. I've used them a couple times in my life, and it worked okay. I mean, some people swear by them. Like I have one lab that has one in their lab, and they just absolutely love it because I think this is very sample dependent if it works or not. It's called an Optigrid, and usually when you hear this, I think some of the other companies have them out now, but Zeiss was always the one that had this thing, the German company Zeiss. Usually had this. It's a little bit different idea, but again, it's a confocal sort of kind of in a way. Okay. So what the heart of this thing is this thing called a grid. Okay. It's going to send light through this grid. So you'll actually get it, and they call this structured illumination. So what you'll actually get is you'll get these things where you see the lines of the grid in it, in the picture. Okay. But if you're able to move that grid in such a way each time that, this doesn't have light, this has like the white, but the black doesn't. But if you're able to move that a little bit each time in a precise manner, and you take 3 different pictures, okay, a lot of physics here, okay, then you'll have the first image will have the lines here, the second image will have the here, and the third will have the, it'll collect this light where it's white, okay. And what you do then is you have focus light, whoops, let's go back. You have focus light coming through, where it's white, where the light's coming through, and not where it's black. And then what you do is you get the out of focus features to blink. The blinking is deconvolved into a confocal image. So basically you're collecting 3 images like this on a grid. You use deconvolution software, a special kind, and it's able to put it all back together. And like I said, some labs, a couple I have work with, they just love the thing. But Optigrid can be fairly cheap. Sometimes Zeiss, like when I used to go against them in sales, I'd be, we'd be going back and forth. I think they're going to buy the Nikon. Then Zeiss would go, well, we'll throw in an Optigrid on the system. And then I'd be like, I'd lose. So sometimes it hurt me. But it can be fairly cheap. It's not really good on thick specimens. It tends to impart artifacts, and it can be very slow because of the computer processing. But I'm thinking this isn't so bad now because the computers are so much better. But it's just a different variation on confocal. I don't know if I'd truly call it confocal, but you get definitely confocal-like images. I mean, I guess you could argue, is it, isn't it? But it's another variation. And it's another cheaper alternative. Okay. And then yesterday we talked about deconvolution, where you collect an image, you have the light above it, the light below it, we run mathematical formula that says, hey, if this was all perfect, what would it look like? So we're getting the light above the focal plane, the light below the focal plane, send it into the math, into the algorithm, which you actually have on your metamorph. You actually have under process, you'll have 3D deconvolution in there. Okay. Now, if you go to buy this for the institution, the one thing you have to keep in mind is that the metamorph I'm giving you has everything. Okay, it has everything. It has absolutely everything they make. But when you buy a setup of metamorph, it's like a la carte. Do you know what a la carte means? A la carte means like if you go to the restaurant, you buy, you buy the potato, you buy the bread, you buy each item separately. So the way they sell metamorph is they give you a little bit of a package, but then they sell you each item separately on top of it, depending on what you need. And my deconvolution I use, just that part alone, like an individual metamorph when I bought it was like 6,000 or 5,000 or something like that. But just the deconvolution was 7,000. Okay. So keep that in mind. We'll talk about sales the last day. It's a different world. Okay. Deconvolution assumes you have a perfect optical microscope system. You collect a Z-stack. like we've been talking about. Remember Z-stack, moving through Z, taking the pictures, getting a deck of cards. Insert the parameters in the software. Usually one of them is the emissive wavelength and an XY calibration, and then you run it into deconvolution algorithm, and woo, you go from this to something like this. You go from this to something like this. Okay, so definitely more confocal-like, and it definitely cleans up the images. Okay, you can see the difference. It's pretty obvious. It's pretty cool when you actually run it and you watch it happen. It's like, wow, that's pretty cool. Okay. So I like it on thinner specimens. A lot of times in my lab, I'll just do it on cells, because cells are thin, and I don't want to go over to confocal. The user doesn't want to spend the money, which I don't blame them. And it's trouble on thicker specimens. It's the kind of thing you almost have to run it to see if it's going to work. And then on your flash drive, I put a good article if you want to read more about it from a company, AutoQuant, wrote an article in that Biotechniques, or not Biotechniques, Biophotonics magazine I mentioned to you that's free. So there's a pretty good article in deconvolution a few years ago, so I just put it in there. It's on your course materials if you want to read more about it. Okay, so 2-photon microscopy, okay? 2-photon microscopy. So everything we've been talking about up to this point, we've been saying you got to push the energy in to make the fluorescent molecule, to make the electrons jump to the next level, for them to come back to make fluorescence, right? So this is what we've been talking about here. We're bringing in an excitation light, just one of them, and we're able to pop it up to this energy, okay? So we're able to use like a blue light to make our green fluorescence work, right? We talked about the change in colors. So we're able to use a blue light here to make our green fluorescence work. But the energy here is pretty high. A blue light has a lot of energy, right? Roy G. Biv, it's down in the Biv part. Okay, so what if instead we used like lower wavelength, less energy photons, but instead of using just one of these, we use two of them. So say the energy here is 100, okay? So let's say the energy here is 100 that we need to make our molecule fluoresce. Well, if we could use one photon of 50 and another photon of 50, it's still going to give us 100, right? We're still going to have enough energy to make this event happen, to excite it. But now, instead of using just one, we use two. Okay, so what's the advantage? Well, the biggest advantage of 2-photon microscopy by far is this. The lower energy photons can travel much deeper into the tissue than can higher energy photons. So you can go way into the tissue. I mean, I've seen people go like 2 millimeters into the tissue, which is just craziness. When you think about all that fat in the way and all the carbohydrates in the way and all the other proteins in the way. But you can actually image that deep and not get all these scatter problems and such using a system like this 2-photon. Now, you used to use this Shrejith, right, with Dr. Wong's lab, I think? Okay. You did it intravitreal, so, okay. But the same idea. So they're out there, they're pretty cool, they're really expensive because the lasers are special lasers. They have to be able to throw 2 photons at the exact same instant for this to work. You can't have one come in 50% and then one come in 50%. They have to both come in. 2-photon. Okay. If photons hit a fluorophore at the same time, their energy can be absorbed simultaneously to produce a fluorescent excitation. That's what we just talked about, right? The energy yield from the 2 photons is equivalent to that of 1 photon of twice the energy, right? 100, 50, 50. So 2 photons of red light can excite a fluorophore that normally requires an ultraviolet illumination. Again, these more destructive, right? Shorter wavelength, more energy. This, less energy, less scattering, could go deeper. Everything's a trade-off. Okay. So, the 2-photon doesn't need a pinhole because of the way it works. Okay. Okay, in laser scanning, the microscope objective focuses the laser beam to a small spot at the tip of a cone. Light travels through this crossover point in the beam and it spreads out again. So laser scanning, remember, you're hitting it like at a point. Everything we saw was like a point diagram. Okay. But in 2-photon, it just matters where the light hits. Because it's only going to excite. It's not going to excite above it. It's not going to excite below it. It's only going to excite where those 2 photons meet. It's not going to excite anywhere else. Because the energy is not enough to excite anything else. So it only excites where the 2 photons hit. That's the main takeaway from this whole slide. Okay. Okay, so basically the take-home message, the result is a sub-micron optical section, but without the necessity of it coming through a pinhole, because you don't need a pinhole because the only place it's going to work and excite something is where it hits. So you have that really tiny area where those 2 photons actually meet each other and hit and excite that one molecule. Okay, so it avoids the pinhole. And here's the way to look at it, is single photon, you're going to get light, I mean the laser light's coming in, and it's going to excite some of this stuff, and then you're going to use the pinhole to just get what's in focus, right? But here, you're only going to excite right here. So you're not going to excite any of this stuff at all. The fluorescence won't fluoresce. But on a regular confocal, it will excite here, and it will excite here. But you're using the pinhole to grab it out. Here, only where those 2 photons come up with the right energy will it work. Okay? That's the difference. Of course, advantage, deep tissue. Other advantage, less photo damage. You're using a longer wavelength, less energy. Okay, so the red light by a 2-photon is less detrimental for living tissues. Okay, now, the maximal spatial resolution is reduced compared to a conventional microscope because the longer wavelengths used use lower numerical aperture 2-photon objectives. Okay, so a little bit of a trade-off, but again, a nice technique if you want to go deep. Okay. Again, the elimination of damage to non-image planes. You're not hurting the areas outside of the plane where you're actually collecting. Okay. And basically, most biological tissues don't absorb at those red and infrared lights. Like I was telling you yesterday about autofluorescence, we try to work more out in the farther out in the red because you get less autofluorescence, less absorption of fluorescence. Okay. So the collateral damage is virtually absent even after prolonged exposure. Okay. Okay, and then photo bleaching, you'll get this kind of photo bleaching with the regular confocal. You have this big cone of area that'll get photo bleached. Whereas here, the 2-photon, the only place you're exciting is right here. So the only place it can bleach is right here. Okay. So this is sort of crazy. This was, I think, 100 microns deep, and you're able to get the nuclei really nice in a myocardium, which is a muscle tissue. Okay. So deep, if deep's your thing, if you have to go deep, this is the way to go. That's the big take-home message with 2-photon. If you want to go fast, spinning disc. If you just want to do a lot of normal kind of stuff, laser scanning. Okay, again, application, application, application. Okay. The disadvantages, though, are it can be hard to figure out how to get things to excite because they can be a little bit difficult. The one thing is, you know, infrared light, like heat is infrared. It tends to heat up your sample a little bit. Not really damage it, but heats it. So it's something you might want to think about. Damage to monolayers might be greater because it actually can damage protein residues. And the biggest thing about 2-photon is you got to be ready to do it because those lasers are ridiculously expensive. And I think a laser, a normal laser here is probably $70,000 laser or so. They're expensive. I might be wrong about that, but they're very expensive. Okay. This is just a comparison of some of the stuff between 2-photon and regular confocal. Some of the things we talked about, just showing you, you know, what the pros and cons are of each. Okay. And like it says here, critical cost, laser care for the 2-photon. Okay. So again, to reiterate where we started today, start with something simple. I mean, if simple works, why go crazy? Why spend the money? Why, you know, why overdo it if you don't have to? So again, I really like wide field for a lot of stuff. Maybe structured illumination. Maybe deconvolution is your answer. Okay, we'll get into some of these other things, turf and these other confocals. But I always try to just, like anytime, even if we're going to go to the confocal, ultimately I always tell people, hey, bring it over to wide field, let's take a look. Let's make sure the experiment worked. At least we can get a handle on if it worked. Why go to the confocal, spend the money, spend the time to get over there to say, oh, it didn't even work. So at least use the wide fields or whatever to get an answer to start with. Okay. And I'd like to thank, for this part of the talk, again, when I went to that workshop in Maine, which I thought was actually fabulous. It's called Qualitative Fluorescence Microscopy. It's run by Simon Watkins. And Claudette St. Croix had a lot to do with it. Both of them got together. They wrote a book, and I used a couple things out of this book that they had. This is an absolutely fabulous book. Okay, like everything I'm talking about over the next few days, whatever, is explained in detail in here. Lots of protocols, lots of other things. It probably wouldn't be bad to grab one of these books for the institution, something like this, because it is just very well written, very clean, very. It's just a really good book, I guess is the way to put it. And it's written by Simon and Claudette St. Croix. Okay, these are both at the University of Pittsburgh too. Both of these, which again we were talking about schools yesterday. For microscopy, working under somebody like this would be, if you really want to be a microscope genius, working under somebody like Simon would be fabulous. And that ends that lecture. Thanks. But speed, I would definitely think of live, the laser or the spinning disc, or deconvolution is another way to go about speed. Because you could take a wide field image, and then later on you could process it to get a little cleaner. So I guess when I say pseudo, I guess I'm talking about like the Optigrid and deconvolution. They're pseudo, sort of, kind of confocal. You can go faster though than the laser scanning.
And if we want to look specifically at particular planes,
yes.
you said we can focus on that area. How can we choose that area specifically in all the cells?
Okay, so what you do when you actually get on the scope is you actually pick what they call a top, like you're focusing on your cell, right? And you focus up and up and up and up until it starts to become blurry. So then you're out of focus. So you call that the top. And then you focus, focus, focus. You see your cell in focus. You see your cell in focus. Then eventually you get to a spot where it's blurry again. The cell's gone.
No, that's not my question. My question is because on the slide there are several cells.
Right.
They're all different directions.
Right.
And the spot which we are looking will be in a different plane.
Okay. But basically what you're going to do is this. Like when you're looking on a slide at cells, okay, like here's your slide, right? And these are your cells. Right? They're only so thick. So what you're going to do is you're going to start your stack here, and you're going to start your stack, maybe stop it here, and then you would just continually go across it, and then you get the top of your cells and the bottom of your cells. So you get your whole cell each time. That's usually how we go about it. That's like I was saying, when you actually physically do it, you look to where it goes out of focus. So you know you're up above it, and then you go down below it out of focus. And then as you go across, take different pictures. If you do that with every cell, you'll get the whole cell each time. That's how we go about it, to try to capture the same thing every time. Because, a little bit of overkill is okay. So you get a little bit of dead space here, a little dead space here. That's fine.
Suppose we are looking at the nucleus specifically.
Yes.
So
same idea. You would focus on the nucleus, and you'd start to move up and see where you're not in focus anymore and stop, and go down where you're not in focus anymore and stop. And you would just then get the nucleus in terms of Z. Because again, if you're just looking at the nucleus, why take all this above it and below it? Just take close to it. You always try to go a little bit above it and a little bit below it.
And you said in the spinning disk, there's a disadvantage, Z
is a little thicker. Right, than the other. But, I mean, if it's live cell spinning disk, the speed probably outweighs that difference in Z. Everything's a trade-off. Nothing's perfect. Any other questions?
What about the cell preparation? Do we always have to fix the cells, or is recommended to?
Okay, well, if it's live cell, you're not going to fix it. Okay, but then yes, like we're going to talk about antibody staining and stuff in the future, and then yes, that would all be fixed. Those cells, that would all be fixed. Okay, and I'll talk when I get to the antibody stuff. I'll talk a little bit about techniques, how to stain it. But again, a book like that, or even if you just get on Google and look up fixation protocols for cells, there's lots of different things out there to tell you how to do it. Does that answer your question? Does it? Okay. Somebody else I saw? Yes.
Is the spectral unmixing, how do we give that pseudo-color to that particular protein? Is it with the help of software?
Oh, the pseudo color?
Yeah.
Yeah, it's as easy as, I could show you in Metamorph today, it's as easy as you just click on a thing, it looks like a rainbow. You just click on it, pick red, or pick green, or pick blue, or pick whatever, and it just assigns the color. It's that easy, it's all in the software. It's just, they call them lookup tables, and the lookup table applies green to the picture, or applies red, or applies yellow, any color you can think of pretty much, you can apply to the image. Because remember, it's not colored, it's just photons, it's just math. Okay. Other questions?
What is the optimum pinhole diameter to get maximum
Okay, the optimum pinhole diameter is determined by what objective you're using and what its numerical aperture is. There's like a formula for that. But like I said, on these confocals these days, you'll put in, I'm going to use this wavelength and I'm going to use this objective. It calculates it for you. So it gives you the optimal pinhole. I mean, it's good to know about it, but the machines these days, they automatically do it for you. So then you're okay with the optimal pinhole because it changes based on numerical aperture and based on the magnification and stuff. I don't know. I think there might be some, but I don't know for certain. I can't answer your question. I really don't know. I think there might be a little bit, but I really don't know. You would just have to look. I don't know the answer to that.
For studying GFP type of protein, which is better, Fluorescent Microscopy or Confocal?
What kind of structures are you looking at?
Bacteria.
Probably confocal. Probably.
What about acrid orange stain, like for Confocal microscope?
Oh, acrid in origin, and you're staining what, like white blood cells or something like that?
No, bacteria.
You're staining, I mean, it depends. I mean, if you want details on the bacteria, you're almost going to have to go to confocal. But if you want just like, hey, the bacteria is here or the bacteria is not here, then it would just be a wide field application probably. So, probably confocal, I would think. I mean, things are dependent, but it sounds like what you're talking about is probably more confocal, probably, I would think, for something like that. Right. Does that answer your question? Okay. Anybody else? Yes.
In a spinning disk, how the disk make the movement of light faster? Because you have shown like it is
Oh.
like this.
Oh, the spinning disc?
Yeah.
Okay, so what it is because, like on the laser scanning, you're taking the pinhole and you're moving it across the image like this. It's sort of slow, like a vacuum, right? Like cleaning the room. Whereas the spinning disc, it lets you put the pinhole on the whole image. So you take the whole image at once. You don't have to go back and forth. It just goes, take the whole picture,
So,
take the whole picture, take the whole picture.
Now, if we consider disk, we will be having a how I cannot call about it.
Okay, so what it is it, because it spins, okay, because it spins, it's got all these holes, right? And it's spinning, it's going around and around. So it puts that on the image. So you have this whole big area that the pinhole's applied to. So it can take the whole image at one time. It doesn't have to go like the laser scanning.
So the light goes.
Yeah, it goes, the light just gets shined on here and the spinning disc generates pinholes across the whole image. Yeah. So so this will take the whole image at one time. So it will take, like let's say this is your sample, right? It'll take, with spinning disc, it'll take that whole thing, it'll take that whole picture at one time. Whereas with laser scanning, it has to go like this. It has to go that, and then 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 that, to get the picture. Whereas this just goes, wham, takes the whole thing at once. Whereas this has to go across it to generate it. So this is much faster because you're taking the whole frame, the whole picture at one time. Whereas here you're taking the picture as a series of scans. Right, exactly. Does that make sense?
Yeah.
Okay. Any other questions? Good questions, too, by the way. Very good questions. It shows everybody's thinking today, which is good.
Thank you, sir, for that interesting talk.